Welcome back, guys. So today we're going to be getting into cell organelles. What the word organelle means is like a little organ. So since cells are the basic unit of life, they have different compartments and functions, sort of like how our bodies do. So it's kind of blows your mind just a little bit when you think like they're just like miniature bodies that make up your body. I probably just messed with a couple of y'all. I'm sorry. Okay, so going back for a second. So if you are taking notes, which you really ought to be, the easiest way to take notes for this section is most likely going to be in a chart format. So you're going to be getting a lot of the names of the pieces and then the function. So you're going to want to keep up with that, okay? Now you think you can do that on paper, that's fine. So however you want to do it, the name and what they do, you are going to be tested on the functions of some of these. And also your activity today is going to involve you labeling, so just be prepared for that. Okay, so the cytoskeleton has many functions. So cyto means cell, and we know what a skeleton is. It's like the bones in your body. So cells have this kind of thing too. So what it actually is, you can see in the image here, they have used a fluorescent stain on the cytoskeleton of the cell. So in biology, what we can do is use special dyes that only attach to specific parts. So if we know the cytoskeleton is mostly protein, we can put a dye that specifically attaches to protein. And then when we look at it under a special microscope, it fluoresces or lights up like how you see in the image. So the cytoskeleton is basically like a big protein net that gives a cell shape and support. It helps position and transport organelles because it's kind of like on the inside as well. It provides strength. It can assist in cell division and in movement. So the closest thing I think of to this, I don't know how familiar you guys are with an octopus or a squid. They have something called a hydrostatic skeleton, which means that they don't have any bones, which you probably knew, but they basically push against the water with their body, and that's what gives them the shape. It's also kind of like a net, a bit. Okay, so moving on to the next one. Ooh, do I have a pointer? Oh, okay, cool. I'm sorry, I just figured something out. Apologies. Okay, so the nucleus stores genetic information, and we learned when we did macromolecules that genetic information is stored as DNA. So in a cell, all the DNA is kept right there in the nucleus. So I'm going to give you an extra term because it's on your sorting activity, and I don't think it's in this presentation. So there is something called the nucleolus, which is that dark purple part. I wish it would let me write on here. It doesn't. Okay, so the nucleolus, it's like nucleus, but it has like an extra syllable in there, is that dark purple region. And that is going to be where we make like RNA. It's like just a specific dark spot in the nucleus. You'll see. Okay, so many processes occur in the endoplasmic reticulum. So that is a big, nasty word. This is an actual photograph of one, which is pretty cool. All these little red guys are telling you are something called a ribosome, which we will talk about in just a second. Okay, so there's two types of endoplasmic reticulum. There is a rough one. Okay, see it's got all these little bumps over it, so it's rough. And there's a smooth one, and see it doesn't have the bumps. Okay, so they transport materials through the cell and they help make proteins and fats. Okay, so the rough ER has ribosomes, so that's going to be where the protein shenanigans is happening. And the smooth ER doesn't have those, so it's not making proteins, it's making fat mostly. So it does help transport material through the cell. This part right here, I don't know if you noticed in the other image, this, if you can see the little red dot circling around, this is where the nucleus is. Generally speaking, the endoplasmic reticulum can help connect the nucleus to the outside of the cell. See, it just goes through these little pipes and it goes through the cell that way. Okay, so ribosomes, you're gonna find these in literally every kind of cell, from bacteria all the way up to cells like ours. They link amino acids to form proteins. When we get to Unit 2, we will talk about that in great detail, about how that happens. But for now, all you need to know, ribosome links amino acids to make proteins. Okay, so they're super small. They're actually just a couple pieces of RNA and protein put together to make like a little thing like this, and you feed the RNA message through and it makes protein. So Golgi bodies, or the Golgi apparatus, you are going to hear it both ways. It just depends on the book and the teacher which term you're going to hear. So they package proteins and stores or ships them out of a cell. So it's going to receive the things that are made by the ribosomes, the endoplasmic reticulum. And so I don't know if you can read this image, but it says that there is cis and trans in science just mean sides, or different sides, okay? So on this one side, it's receiving incoming vesicles or containers of stuff. It may process them, maybe it has to put a finishing touch on them, then it repackages them and it sends them out. Think of it like Amazon. They get a lot, they don't make any materials in there necessarily, but they receive shipments of things in from the factories. They repackage them and then they ship them out wherever they need to go. That's what the Golgi body, or the Golgi apparatus, is doing. And it's actually named after an Italian man whose last name was Golgi, and he discovered these. Okay, so a vesicle, whenever we talk about that in biology, is just a membrane-bound sac that holds materials. That's all it is. If the cell needs to store something or bag something up, basically, they're like the little to-go containers of a cell. So as you see, there's like a ton of them, especially if your cell is one that produces something like a hormone or an enzyme or something. It might make a ton of these little guys. So the Golgi apparatus has been super duper busy. It's made a whole bunch of these and they're getting ready to be pushed out of the cell. So mitochondria, I'm sure I heard that word and there's like about 10 of you that just like, mitochondria is the powerhouse of the cell. So that definition, while it might help you remember it, that is not the definition we're going to use for our purposes. So mitochondria supply energy to the cell. That's the basic thing. A little more detailed is they're breaking sugar down into something called ATP. So that's how ATP is like the molecule, it's the universal energy currency. So all cells except ATP, they know what that is. And the mitochondria is able to break down the sugars we eat into that energy, into that ATP. Okay, vacuoles. So these are a fluid-filled sac that holds material. This is an actual photograph of a plant cell. This big light-colored area here, that's the big central vacuole. We're going to talk in a second about how that's actually one of the key traits that you know this is a plant cell, not an animal cell, is plant cells have huge water storage containers in them. Huge. Animal cells don't do this. We still have vacuoles. The word vacuole just refers to empty, like the vacuum of space is empty. There's not a lot there. If someone is vacuous, like their brain's empty, okay, so a vacuole is an empty bag. It looks like an empty space when you look at a slide, but it's actually filled with liquid, okay? So lysosomes contain enzymes to digest material. I think like Lysol, they're like the cleanup crew of a cell. If something wears out or dies or whatever, they've got these digestive juices, these enzymes that they can go over and they can break it down. They clean it up, okay? So centrioles, they're these little cylinder looking dudes over here. they actually help cells divide because they're helping to divide the DNA. I think of it like a reel on a fishing pole. So what happens is they attach little protein cables basically to the different ends of a chromosome, and then they reel them in and they break them apart. So that's what centrioles do. They can also form cilia and flagella. We haven't talked about that a ton. What a cilia is, whenever you see a cell and it's got like little hairy projections off the side that it can move around to make it move, those are cilia. A flagella, it looks like a tadpole's tail. Sperm have them. A lot of free-living bacteria do as well. It's a whip-like tail they can use for movement. So that's also made from centrioles. Okay, so cell walls and chloroplasts. These are two things that are only found in plant cells for our purposes. So a cell wall provides rigid support. This is an actual slide of some plant cells. So instead of being like a blobby shape, like the cell on the start of our presentation was, you know, the one looks like a booger. This don't look like a booger. This looks like, I don't know, a little, I hate to say it, it's like a cell. This is the kind of cell that they name cells for, because they've got these rigid little walls on them. So the cell wall does that. Chloroplasts, this is where photosynthesis happens. Okay, they convert solar energy to chemical energy, or they make sugar there. So the chloroplast makes the sugar, and then the mitochondria breaks the sugar down. Plants have both of those, because plants got to eat too, okay? It's just they make the sugar themselves. Our cells, like animal cells, they don't have that. And in a second, I'm going to try to show you the diagrams on my wall back here, because my sister, the artist, went to great pains to draw those for me, and they're an excellent visual aid for this. Okay, so these differences right here, I 100% guarantee you're going to see it on your unit tests. So write it down, put stars by it, whatever you got to do. So the three main differences between plant cells and animal cells, how we're going to tell them apart. Plant cells have a cell wall, okay? So the cell diagrams you're gonna see, these round blobby ones, those are animal cells. These ones with the hard sides like this, those are plant cells. They're usually more angular, okay? So the large central vacuole, this big water balloon basically in the middle, that's what we're talking about. And then chloroplasts. It'll be green, it'll have these disc-like little things in it. That's how you know it's chloroplast. Okay, so those three things are our main differences. I just want to, let's see if I can show you this real quick. Okay, oh, going this way. All right, I don't know how well you can see that. So you've got a plant cell and I've got an animal cell right there. You can tell the differences just by looking at them. You can see our large central vacuole up there, and you see that it also has most of the same organelles as the animal cell down here. It's got most of the same stuff. It's just those three differences we just talked about are going to be only in plant cells. Okay, so let me show you your assignment real quick. Hold on. Sorry, shake the cam, shake the cam. Okay. So I'm going to show you your assignment real quick, and I'm going to explain to you how to work it, because that's usually where we have the most confusion. You're going to let me do this? Exit. Okay, cool. All right. So when you are in your activity for today, you don't want to be in presentation mode. So where it says present up here, you don't want to click that. All right, so please don't. So your directions say while in editing mode, which is what I'm doing. So that means I can click through the slides right here, and I can do stuff to them. left click each label and drag them to the correct organelle on each diagram. When you're finished, submit your work on Google Classroom. It is super important you make sure you turn your work in. So this diagram will be useful to you when you're studying. You are going to have to be able to label a basic cell diagram with your basic organelles. So make sure that you take this seriously and do your best. Okay, so we're looking at this. I'm going to go ahead and give you a couple of these, especially lysosome, because there's a lot of sac-like organelles in this diagram, and you may not, you know, you may not be able to tell. I certainly couldn't tell you what's the difference between the blue circle and the red circle, so we're not super concerned. So I left clicked and it brought up this box. So when I have the little 4-way arrows, what I can do is I can drag this box. See, I'm dragging it across the screen. So I'm just gonna say that 10, that's a lysosome. That's good for me. So you see, I dragged the label over there and I labeled it. We'll do the nucleus or nucleolus, that one's weird. So I left clicked on the word. I got the little 4-way arrow and I'm gonna drag it and I'm gonna put it over where the nucleolus is. Okay? It's gonna work the same exact way down here on the plant cell slide. So I left click and then I drag it to where it goes while holding down. Okay, I know on a Chromebook you gotta do the 2-finger click or whatever, but you just click and hold down and drag it. Okay? So once you've got it all labeled, you're going to need to turn that into me. That is your assignment for today. If you have any questions, please let me know. Remember that we are getting toward the end of our unit. We're getting there, about halfway through. We still got some things to talk about, but make sure that we're getting our cell things figured out. You're going to be getting a project soon about this. So make sure you're doing your best to figure this out because, you know, project time. All right, guys, I'll see you in the next video. Take care.